As we finish up chapter 6, just talking more about some of the other DNA like plasmids and that horizontal gene transfer and some of the other aspects of translation. So a little bit just to tie us in here to the COVID-19 vaccine, because it completely utilizes the central dogma. So the COVID vaccine is an mRNA vaccine. So what is happening is that piece of mRNA that codes for a spike protein on the virus is either put into an empty viral vector that then can bind to our cells to be inserted, or it can be packaged into a lipid vesicle. So we talked about those two options whenever we covered vaccines earlier in the semester. So what's going to happen here is the mRNA is going to be isolated. And again, that mRNA is used to make one of the spike proteins on the COVID-19 coronavirus. So that messenger RNA is then packaged again, either into one of those lipid nanoparticles, or it could be put into a vector. This example, though, it's putting the mRNA into nanoparticles. That's injected. That's what we receive whenever we get the COVID vaccine. So that those nanoparticles are going to allow it to enter into our cells, so through our cell membrane. And then that messenger RNA that we see here then attaches to one of the ribosomes. It doesn't know that it's viral RNA, so it starts making that protein. And that protein then gets inserted into our membranes, and it's the spike protein that's normally on the coronavirus. Well, our immune system is going to recognize the spike protein as being foreign, and it mounts a response. All right, so now we're going to have those memory cells to that, but we were never actually infected with the COVID, with the coronavirus itself. Just messenger RNA that then instructed our cells to make this spike protein to create an immune response to give us immunity to it. So it's a really cool new approach to vaccination. So just a little bit here as we finish up transcription and translation with some of the differences. So we focused just mainly on prokaryotic transcription and translation. Eukaryotic is just a little bit different. Still the main steps: transcription makes RNA, translation makes the protein. Again, that co-translation, though, where as soon as transcription starts, a ribosome can attach and start making protein immediately. Again, that's only in prokaryotes, so only in bacteria and eukarya. Again, in eukaryotic cells, transcription's in the nucleus. That's completely separate from translation in the cytoplasm where the ribosomes are. And then also the AUG is still a start codon, but the amino acid methionine is just slightly different in prokaryotic and eukaryotic cells, but it's still a methionine. And then eukaryotic messenger RNA only codes for one protein, whereas in bacteria, they can have operons or a series of several genes that are all linked together under the same promoter. So multiple proteins can be made from this really long messenger RNA. So it combines different genes onto one mRNA. Again, only found in prokaryotic cells. And then one last difference is in eukaryotes, our DNA has both introns and exons. Prokaryotic cells do not have any introns. And introns, as it says here, are just intervening sequences. So here in our picture, so exons are in yellow, introns are in green. And essentially the intron is just kind of some spacer DNA. It's not actually used to make the protein, but it gets transcribed. So transcription makes mRNA that has both exons and introns. But before it leaves to go out to the ribosome, we need to splice or remove all of the introns out of that message. So there are things called spliceosomes and ribozymes that help do this. So it's the process in which all of those introns get spliced out so that once the messenger RNA is mature and ready to go, it only has exons in it. There are no introns. But again, this only happens in eukaryotic cells, not prokaryotic. Also talking here about DNA, about a process called recombination. So as the name implies, it's combining different things in a different way. So recombination is an event in which one bacteria can donate DNA to another, so that by the end, we now have a strain that's different from either the donor or the recipient because it has this mixture of DNA from two different sources. So for example, plasmids are those extra chromosomal DNAs that are great for moving between cells. So if one cell, let's say we have an E. coli, and it picks up a plasmid from a Staph aureus bacteria, now it is considered a recombinant organism because it has both E. coli and Staph aureus DNA. So it's not just like E. coli, it's not just like Staph aureus, it's a whole new kind of organism or a subset of that. So that is referred to as a recombinant. Other ways of changing the genome within bacteria, especially, is horizontal gene transfer. And we've talked about this in the past whenever we talked about some of our structures. But horizontal gene transfer is transferring of DNA from one organism so that it acquires new genes that did not come directly from the parents. So again, vertical would be DNA coming from a parent. Horizontal is whenever it's coming from another organism other than their parent. And so eukaryotic organisms also can engage a little bit in this, but it has to be aided by microbes like viruses. So we'll kind of come back to that. But for the most part, bacteria are those that are undergoing horizontal gene transfer. So in order to do that, we come back to those plasmids we've mentioned several times. Again, those are just small circular pieces of DNA. Again, they can replicate independent. They're separate from the chromosome, but these can also be transferred between DNA cells. And most bacteria and even some fungi have plasmids. And they can contain, a few dozen genes on them that make proteins, again, unique, that is not required for the cell to survive, but does carry those useful traits. So there are three main types of horizontal gene transfer. So we have conjugation, and this is where there is direct contact to where there's a plasmid called the F plasmid or the fertility plasmid, and essentially one of the strands of DNA is transferred through the sex pilus to another bacterial cell. So it's literally transferring the DNA through that sex pilus. And again, it allows genes like drug resistance, toxin production. It can help make new enzymes. It could be a new adhesion molecule. It can make an enzyme that helps take up iron. It could be multiple things and advantages that it provides. Another way is transformation. And this is where some bacteria, not all bacteria can do this. This is where bacteria can take up actually free DNA. So if we think about being inside of a pond or inside of an area and one bacterial cell just exploded and it released all of its DNA into the water, some bacteria have the ability of just sucking up naked or free DNA. Again, not all cells can do that, but some of them can. So they can, again, bring different genes that help do things like make a capsule. And again, it also helps us in the lab to make different cloning techniques and for research. And then finally, we have transduction. And this is where a method that can actually change eukaryotic cells as well. But in this case, the donor is lysed bacterial cells, and this is using a bacteriophage. So there's viruses that infect bacteria. And essentially what they're doing is they are taking in DNA and transferring it inside of that virus. All right, so just going through each of these a little bit more. Again, conjugation is essentially bacterial sex. Again, there's direct contact that has to be made. So it is a direct mode of transmission to where, again, that plasmid is transferred using that sex pilus. And this can be in either gram-positive or gram-negative bacteria. And again, most of them have this F plasmid or an F factor in them, and especially the most understood is in E. coli for how that happens. And this is just showing that. So in this case, again, we have a plasmid here. It is an F+, so it has this fertility plasmid on it. So it is going to join through the sex pilus. So again, the pilus is just a short projection that sticks from the cell. So two different bacteria join together using that sex pilus. It creates this bridge, and then one strand of that plasmid is going to enter through the sex pilus into the other cell. And in this case, it didn't have the plasmid already, but now it does. And then it just puts the other strand of DNA back using almost like it just went through replication. All right, now these resistance plasmids or factors, as the name implies, they have genes that carry resistance to antibiotics or other drugs. And again, a lot of them are commonly transferred using conjugation. And it can lead to this multiple drug resistance like MRSA and VRSA, to where, again, we have Staph aureus that is resistant to a lot of bacterias because of these resistant plasmids. Now in transformation, again, this is taking DNA in from a solution. So in this case, we have a bacterial cell, and whatever the reason, it lysed, and now it just has a bunch of chomped up pieces of DNA that were released. And here we have another cell that is intact, and it has the ability to just essentially suck up that DNA. And as long as everything goes okay, it gets added to the bacterial chromosome, or it could be a plasmid as well. But now this bacterial cell is recombinant. It has a new gene that it didn't get from its parents. It was able to take it up from the environment. And those cells that can do that are called competent cells. So again, not all cells can do that. In order to do that, they have to have their own genetic makeup that allows them to take in that DNA. Then transduction, this is kind of piggyback DNA. So this is where we're using bacteriophages, viruses that infect bacteria, to actually carry DNA from one donor to a recipient. Now understand, in transduction, the virus, that bacteriophage is not meaning to do this. This is an accidental process that occurs. And so this can occur in a lot of different bacteria, again, because it depends on which bacteriophage is impacting it or infecting it. So that participating bacteria in the transduction event, it does have to be the same species, because again, bacteriophages bind to specific receptors. So this couldn't take like DNA from an E. coli and put it into Staph aureus. Those are two different. The bacteria, that virus wouldn't be able to attach to both of them. So it has to be the same species, because that's how bacteriophages are able to attach, is based on their receptors, and that's different for each organism. Now there are two different versions of transduction, generalized and specialized. So in generalized, what happens is there are random fragments of the host DNA. So just to go over a typical bacteriophage infection. So here's our bacterial cell, here's our phage, our bacteriophage. So it just attached and inserted its viral DNA or RNA. It takes over the host cell by chopping everything up into little bits, and now it needs to assemble. So it's going to put its spikes together in its capsid. And what happens is, again, completely accidental. This bacteriophage is going to take in a piece of host DNA, so a piece of the bacterial DNA instead of its viral DNA. So again, it's not meaning to do this. This is not directed. It's not intentional. The bacteriophage is just trying to produce more viruses, but it accidentally adds bacterial DNA instead of viral DNA. Well, now it's going to exit from this just like it would in a viral infection. But now whenever that phage goes and binds to a new bacterial cell, it's adding in bacterial DNA, okay, that came from the original infection. So now it can get integrated into a plasmid or into the chromosome, and now we just gave a new set of genes to this bacterial cell. So again, in this situation, that phage is just kind of a carrier. It's a bus for it to get to a new bacterial cell. In specialized transduction, it's still the same approach. We're still using a bacteriophage to transfer DNA. It's just a little bit more specific than what general is, because this is whenever we have those prophages. So remember, in bacteriophages, sometimes the DNA just goes in and they chomp it up into little bits and pieces all over the place and then assemble it like we had just seen. But sometimes the viral DNA becomes part of the host genome and it gets integrated, and that's called a prophage. Well, what happens in that case now, if that occurred and there's a prophage and now a bacteriophage comes in and it chomps up the DNA, it essentially excises or takes that viral DNA back out that it just integrated. But what can happen is that pieces like beside of that, so if this is our viral DNA, whenever it comes out, sometimes it's going to bring along some pieces of the bacterial DNA on either side, and that could have genes that could then be added onto a new host. Another way of changing the genome is using what are called transposable elements. And these are essentially weird jumping genes, and they can actually move from one part of the genome to another. So it can go from one chromosome part to another part of the chromosome. It can move from a chromosome to a plasmid, or it can move from a plasmid to a chromosome. So again, it's just jumping from one place to another. And these are commonly found, especially in different cells, including our own. Actually, a large part of our genome, our DNA, is made up of these jumping genes, but viruses have them as well. But whenever they move, it can kind of change how they behave and can change the phenotype of the organism. So we have what are referred to as pathogenicity islands, PAIs, and these have multiple genes that are coordinated to create a new trait, so making it pathogenic. So again, when we're talking about horizontal gene transfer, we're talking about transposable elements. A pathogenicity island would be whenever genes are added that allow it to become pathogenic. So we have a cell that wasn't pathogenic, but after horizontal gene transfer or after some event, it now became pathogenic. And so, for example, Yersinia pestis, its ability to scavenge and get iron was due to gaining genetics or through horizontal gene transfer. The ability of Staph aureus to make exotoxins, that is due to horizontal gene transfer. It was able to take in and create new phenotypes through these events. And so these islands of genes, these multiple genes, are flanked that look like genes that are used to make other enzymes. And then we're also going to talk about changing the genetic code through mutation. So again, there are ways of gaining new information. Now we're talking about some changes that can occur that can also change the phenotype. So first of all, just a mutation is any change to the nucleotide sequence. So if it was ACG and now there's an event, then now we have AGG. We just changed the nucleotide sequence, so that's a mutation. And this is the driving force behind change and evolution, and why all of our organisms look differently is multiple mutations over time. In microorganisms, mutations are really evident because, again, like they're single cells. So they're not affecting a mutation in one cell changes the whole organism. It's not like a mutation in us where one cell gets mutated, but everything else is okay. Again, these are single-celled organisms, so mutations can have much larger impacts. So where do these mutations come from? So there are spontaneous mutations that just randomly change the DNA, and there are induced mutations. And this is from exposure to known mutagens, which are chemicals or physical events that are known to cause mutations. So things like X-rays or gamma rays or various toxins that are used. So inducing them versus spontaneously occurring. There are multiple types of mutations. So point mutations, These are small mutations where only a single base on a gene gets changed. So again, a C changing to a G. So this could also include adding a nucleotide, deleting it, or again, just changing it or substituting it. And then these mutations can be lethal, of course, meaning to a dysfunction or death of that cell, or they can also be neutral to where that change doesn't help it and it doesn't hurt it. Now, these types of categories, especially point mutations, we have missense mutation. That's where a different amino acid is going to be encountered, and we'll see some examples coming up. Nonsense, this is where a normal codon is changed to a stop. So now we've ended translation early because that stop codon was much sooner than it should have been. We can also have silent mutations, and this is where there is a change in the nucleotide sequence of DNA, but the amino acid stays the same. And this is due to that redundancy of the code to where multiple codons bring in the same amino acid. And then we also have a back mutation. This is where the gene undergoes a mutation that reverses to its original base composition. So there was, say, a missense mutation, but now something changed and it mutated back to what it originally was. So that's referred to as a back mutation. All right, so here's just showing some examples of these. So here's our normal DNA sequence. Here's our normal RNA sequence. So using, again, A pair, T pairs to A pairs to U during transcription. Then we separate these into our codons, into our groups of 3. We look these up in the genetic code, and we know our protein should be methionine, threonine, aspartate, glutamate, methionine, lysine, and on and on. All right, so just then to look at some of these different mutations. So in a missense mutation, this is where, again, one nucleotide. So in this case, this was supposed to be a G. So again, this is our original, is supposed to be a G, but now it's a T. So now whenever we transcribe it into RNA, now there's an A that gets added. And instead of the codon being GAC that we have up here, now it's GAA. And when we look that up in the genetic code, we'll see that it's going to bring in another glutamate instead of aspartate. So that's changing one of the amino acids in this sequence. Now we can also see a silent mutation here. So we're, in this case, it should be C, we have CTA. Let me just get my eraser here, get rid of some of this. So here we have a mutation at the same spot. So we're changing this third nucleotide, but now instead of changing this G to a T, we just changed it to an A. So again, a spontaneous mutation, this A is now here. So now we're going to bring in a U in our RNA. So we have the codon GAU. Again, it's supposed to be GAC. Well, if you look up GAU in the genetic code, you see that it brings in aspartate. Well, that's what it was originally with GAC. So there was a mutation, a change in the DNA that changed the RNA, but the protein's the same. So we say that this is a silent mutation. Now, more significantly, we can have what's called a frame shift mutation. And this is where we get insertions or deletions of one or more bases in that new DNA strand. So during replication, whenever we're adding and making our new strand of DNA, so mistakes are made that add extra or skip over some of the nucleotides. So this shifts what's called the reading frame, the separation of codons into groups of 3. So this almost always leads to a non-functional protein. It's not possible to have a silent mutation where it doesn't really change anything in most cases. So these are typically going to be much more severe because every amino acid after that addition or insertion, so that mutation, is likely going to be different from the original DNA. So we're not just changing like one amino acid, we're changing many, many, many of them. And this is just showing an example, again, our same RNA and protein that we had looked at earlier. But now what's going to happen is that we're going to get, and we get this insertion that now is changing all of these nucleotides, and therefore it's going to change all the amino acids afterwards. And in this case, what happens is not only are we changing these two amino acids that should be aspartate and glutamate, we also added in a premature stop codon. So now not only do we have multiple changes, but we also have a shorter protein than what it should have been. This is another example here of an insertion, just a slight different one. We still get another premature stop, but we have different amino acids that are being added here, depending on which is getting inserted. But again, what's happening is not only are we changing one amino acid, but now we've shifted the reading frame. So all of the codons after that mutation are going to be different. And then finally, just looking at the effects of these mutations. So regardless of what they are, many of these, especially in prokaryotic cells, are not repaired. Now we have a repair mechanism, which is why if we have x-rays or we have UV exposure, our cells can repair any damage that we get. But bacterial cells, and especially viruses, do not really undergo that repair mechanism. So therefore, those mutations are permanent, and they're also passed on to their children. So all the offspring after that mutation are going to receive that mutated version of the DNA. So that becomes a big part of the overall gene pool for that organism. And then so a small number of these changes or mutations can contribute to the success of that population. So especially thinking about antibiotic resistance. So if antibiotic resistance occurs in one cell and then it divides and produces, offspring that then produce more offspring. So now we have this population of mutated bacteria that are resistant to an antibiotic. And now if that antibiotic is introduced, it now knocks out and wipes out all the other bacteria. So now that population is what's going to be successful and kind of repopulate the area. So those are very important. So while sometimes the mutations are harmful and kill the organism or are silent, there can be positive impacts to those mutations.